So good morning, everyone. Just want to first thank Dr. Clark and also organizing a committee just for the invitation. So my name is Joshua Peeples. Feel free to call me Josh. I'm an assistant professor at Texas A&M University in the Department of Electrical and Computer Engineering. So as you can suspect, my background is definitely more in the EE side of things and computer science. And so recently our group has gotten into working with plant phenotyping, so decided to present that work here. I'm also happy I followed NOAA because we actually leverage a lot of our plant CV for our work that I'll mention during the presentation. So a little bit about myself. So I'm originally from Alabama. You might hear a little accent from my voice. So I did electrical engineering for my bachelor's at the University of Alabama at Birmingham. Did my graduate work at the University of Florida, so go Gators. And now I'm a currently assistant professor, started about 2 years ago, fall 2022. Research interest, machine learning, computer vision, image processing. Also, fun fact I usually like to tell people is I have a twin brother. We're big football fans. Usually we'll be at a college or a football game or something once a year. And so I'm also the director or PI. My group is the Advanced Vision and Learning Lab. We're focused on like a lot of algorithm development for machine learning and computer vision. And so from that, when I was at the University of Florida, my research focused mostly looked at this area of texture analysis at the top. And so for those who aren't familiar, texture is something that's easily understood by us as humans, but how do you integrate it within the algorithm? And so, for example, if you all were to touch your desk in front of you, you would say it's a smooth surface. And so the question is, how do you integrate that within different algorithms? And so the reason this is important that depending on application that's being studied, texture can take on different meanings. And so during my PhD, I did a lot of work with the Office of Naval Research for automatic target recognition. And so texture took on different meaning there as opposed to texture in plants. Even though texture can take on different meanings, it's useful across a variety of different tasks is what I looked at a lot during my PhD. So from there, we expanded a bit. And so we've done some multi-scale analysis, so related to what Noah mentioned in his presentation, also explainable AI, and also have some work looking at change detection or anomaly detection for Sandia National Lab currently. And so, of course, I can't do the work by myself, so I have a great group of students that helps me across the different projects. And particularly, I'm going to focus on the phenotyping aspect today. Students that helped out with that effort would be Akshata, Prathamesh, Shri, and Amar, with the asterisk shown there, as well as the undergraduate student, Michael Morris, has been helping out with this as well. All right, so getting into what I want to focus on for this talk. And so Nara did a great job explaining what plant phenotyping is. And also, just when you think about plant phenotyping, there are a lot of key applications, right? So you think about food security, energy demands, biofuel. So a big takeaway from the slide is that phenotyping is important. And so it's important for us to look at this concept in this area of study. And so at Texas A&M, we have a new phenotyping facility that collects high-resolution images of plants. And so I definitely want to mention one of my mentors and collaborators, Dr. Seth Murray, connected me with this opportunity at A&M. And we've been working together to develop this end-to-end data analysis pipeline. Where we break it up into 4 different tasks, and I'll walk through each module as we go throughout the slide, where we start with the data collection all the way to this process called data discovery with a GUI. And so the first part gets into data collection. And so just to give a little bit about the facility, if y'all ever get a chance to come to Texas A&M, I encourage you to go tour the facility. And so with this facility that we have, we're able to collect high-resolution images of plants across multispectral information. I'll show an example next slide. So how the system works is that we have a robotic arm that can take both side scan and top scans. For this presentation, I'm just going to focus on side scans, but you can also get multi-aspect information from the plants, which is important to see. And so from there, like I mentioned before, and also Noah mentioned during his talk, the facility collects multispectral images. So instead of just a typical RGB, we can collect 4 different spectral bands. And so that ranges from green, red edge, red, and also near infrared, which was mentioned before. And so this is an example. Just looking at it, looks like your typical grayscale image, but it's captured across different bands what we're looking at with the plants. And so from that, we recently had an accepted paper to IEEE, which is my professional society international conference on machine learning applications, where we published a subset of this data that we've been collected. So for those who are interested, I'll have a QR code. You can feel free to use this data set. It's available for classification, object detection, segmentation. And so again, this is subset data we've been looking at, but we have about roughly 1,100 images, 512 by 512, captured across 3 different crops, corn, cotton, and rice. And I think someone mentioned before about creating like a pseudo RGB. And so for this data set, all 4 spectral bands are available, but we primarily use the green, red, and red edge to create these like pseudo RGB images for processing with different pre-trained machine learning models. And of course, you can expand data set. So we added the capability to add data augmentation, so like cropping, rotation, different translations like this. And so again, this is publicly available for people to use as well. So the data set I showed you all, it took probably about like a year and a half to 2 years to collect because we had like a lot of different obstacles that we're coming doing like pre-processing. So if you talk to any machine learning person, the biggest thing is cleaning and getting your data prepared, right? So we spent a lot of time looking at that. And so with this portion of the module, on the top, you'll see an image that we had before where we had to look at different normalization techniques. So for those familiar, you got like min-max normalization, standardization, different things like that. We tried a plethora of different techniques to try and clean up the data. And also one thing you'll note here, might be difficult seeing the audience here, is that also, even though we're collecting high resolution images of the plant, we still have some kind of artifacts or background from the facility that we need to be able to account for. And so one of my students looked at this for their master's thesis, and so I'll present some of that work here today as well. And so like I mentioned here, we use traditional computer vision techniques like Noah mentioned before, and so we started using more of these machine learning-based approaches to help improve the process. Okay. And so right now, our pre-processing workflow has 2 major steps. So we have object detection, so detect where the plant is in the image, and then we do a further segmentation step to further isolate it from the background. And so what my student focused on for the master's thesis was this object detection. And so for those familiar with this, a very popular model, you only look once. Yellow model is used a lot for object detection. And so we adjusted it for plant detection. And so even with the yellow model, it's a pretty robust model that people use a lot, but it can have difficulty with detecting like small objects, for example, or operating cluttered scenes. And so one thing that we looked at was, could we improve the pre-processing of this model through something called a spatial transformer network, STN for short. And so here's an overview of the framework that we had developed. And so we have our image, right? And then object detection, for those who aren't familiar with this, basically you want to find like a bounding box located on your object of interest. And so we had the yellow network that has a complex structure with different convolutional layers, so things that process spatial information, for those who aren't familiar with that. And so we have this portion of this framework called this STN, which I'll detail on the next slide, just so it's clear. So space transformer networks were introduced, I think, around like 2015. And so basically this is a special type of artificial neural networks that helps improve spatial invariance in your images. And so the good thing with this method is that you can integrate it within any artificial neural network. So you're not restricted to, say, CNNs, for example. You can integrate it with transformers as well. And so, again, the focus of this is trying to improve robustness to different spatial invariances that may be countered. And so, for example, say we pass an image that has like, the plant might grow, some way that might be complex for the algorithm to detect. Can we learn some kind of transformation? So think about like rotation, translation, scaling, so affine transformation, what we call them, and we try and correct it in the model to improve the detection process. And so my student looked at this again for their thesis, and so what we found was that we were actually able to improve the identification of the plant as opposed to the baseline model. And so this image that you are looking at here is an example of a cotton image that was collected in the facility. And so here, the images on the far right show like the importance that's being detected. So ideally, you want all the warm colors or brighter colors to be indicated on top of the plant as opposed to like the background or pot. And so the baseline STN yellow, or the baseline yellow model, what we found was not able to sufficiently detect the plants. As you can see, it's really paying attention to the pot, which is really isn't what we care about, right? But we're able to show with our approach, it can help identify the plant, which is the most important part of the image. And so we showed an improvement both qualitatively and quantitatively. Again, this work will be presented in December at a IEEE ICMLA conference. And so code's available, data set's available. I encourage y'all to look at that as well. So after we're able to identify the plant, right, with object detection, another aspect is that with this facility, we can collect multi aspects of the plant. And so I remember mentioning we have both like top scans and side scans. And so this is an example of what can be collected in the facility is that we have multiple different looks to the plant from the side. And so even though we have one plant, we can capture, say, like 8 images at different plant heights. And so the idea is that you don't want to just individually look at each aspect of the plant, but you want to analyze the entire plant. And so to make this a bit more concrete, how many people are taking like a panoramic picture on their phone? Great, so it's kind of like the same thing, right? You're just basically taking an image of the plant and you want to stitch those different views together. So some, related to like the orthomosaic that Noah and Seth was mentioning here, but we're just trying to stitch the image together as like a panoramic. And so this is an example. So one algorithm, we looked at several different algorithms. The one we're using here is something called ORB. And so now we're able to, okay, instead of just looking at each individual scene, let's fuse it together to analyze the plant all at once, just so we get a holistic view of the plant. And so this is all that we're showing here. It's this image stitching result. Also, if anyone has a question, feel free to jump in and interrupt as well. Yeah. All right, so we got from data collection. Yeah, go ahead. Yep. Yeah. Mm-hmm.
Thanks. So what's the advantage here? Like the looking at sort of image number 2 and the final image look very similar to me. So what extra data do you get here? And what's the advantage there?
Yeah, great question. So this example, to your point, it's really dependent on the plant. So this isn't the best example to illustrate that, we might need more information. Because some plants we looked at, they kind of like overgrow the pots. And this example is, to your point, like most of the plants contained within the pot doesn't really like grow out, right? But you can imagine if you have some growth that's like very low and it's very hard to capture, you want to be able to get like a holistic view of it. Does that make sense? But yeah, to your point, this isn't the best example to illustrate it, but hopefully want to get like a full picture of the plant. So, yeah. So, great. So we go from data collection, right? So we have like these multispectral images. We do a lot of pre-processing, so using like computer vision, machine learning, different techniques like this. Now, next step, which will lead into our use of plant CV, is once we have this cleaned up representation, what do we do with the plant? Okay. And so just to recap what we do, we get the stitched image, do the bounding box detection, and then we do segmentation to isolate it from the background. And so from there, we leverage Plant CV to extract different features. And so Noah did a great job explaining this. And so these are some of the features that we looked at in the facility, such as the shape of the plant, height of the plant, different things like this, that can be used for different downstream tasks like classification, clustering, anything that you want to look at in terms of machine learning. And so also a good thing with this I really like about Plant CV is that, we get these numeric values, but we can also visualize what's being captured here, right? Because it's important to establish trust between end users of our system. And so it's definitely been a big help leveraging the Plant CV framework for like our phenotypic feature extraction process. So, and Noah alluded to this as well in this talk, is that with the measurements, we get these in terms of pixels, right? But this pixel really makes sense to someone who, is in the field. They don't really care about, oh, this is like the height in pixels. So one thing that our group had to work on was trying to convert these pixel measurements into some kind of physical quantity, like centimeters, for example. And so with the robotic arm, we needed to get some more information. So we had to reach out to the company to get this information, to where typically this robotic arm, as y'all can see in this picture, is kept at like the standard distance between 350 to 400 millimeters, for example. And so with this, with the facility, you can control how many looks you have at the plant. So say a user wants to take, 8 images of the plant or 16. This is something that you, as an end user, decide how many times you want to look at the plant. And so in collaboration with Dr. Seth Murray's group, we did an experiment to where we wanted to see if the system could collect some kind of metadata, which we could. And so with Dr. Murray's experiments, one thing we also want to keep track of is, what plant we're looking at. And so we have some metadata that's collected from the system, but also we verified it with Dr. Murray's experiments, where we have some kind of barcode information to keep track of, we're looking at plant 1 versus plant 10, for example. And so this is a way for us to cross-check what's being collected by the system matches what like a user does for their experiments in the facility. And so with the facility, we can collect different metadata that I talked about. So we can get the position of the robot, position of the plant, and you can actually use these calculations to convert your pixel measurements to some kind of physical quantity. And so I think I showed you all this example before where we have like the different aspects of the plant, right? So going from like 1 to 8, so going like from the bottom to the top. And so we're able to use this to get some kind of physical measurement as we see on the next slide. And so just to make it a bit more concrete, so for example, we can measure the plant height in terms of pixels, but we need to convert it to some kind of physical measurement, right? And so a good question was asked after Noah's presentation about repeatability, right? And so one thing we're working on now is we have an end user in the facility that actually went and got actual measurements of like the plants that they're looking at. And so right now, ongoing work is we're trying to cross-check what the software estimates versus what like an end user estimates to see if we can get some kind of error measurement on like our different physical traits we're looking at. Okay. All right, and so in addition to plant statistics, again, I'm a computer vision person, so I look at image features a lot. And so if you look at the literature for images, there are 3 categories of features typically. There's going to be like your color information, so we talked about like the RGB, like spectral information. You have shape and also texture, which is what I focused on a lot during my PhD. And so texture is really important because typically, you know, color and shape isn't always like the most informative features, and so a lot of people look at this area of texture analysis. And so within the facility, we integrated different texture-based features in these examples that we looked at. So we looked at the HOG, histogram-oriented gradient features, SIFT, scale invariant feature transform, as well as local binary patterns. So these are very common like texture features that people look at for a variety of different applications. So one aspect of texture that's important, as y'all can imagine, is the scale that you look at with the textures. For example, so if y'all think about an image that you take, an object can look very different if you zoom in or zoom out, right? And so that's something that a lot of people look at is how can you make sure your texture measurements are robust across different scales that you look at the image. And so in this example, we took an image of a leaf and we computed this lambda value, which I mentioned is a lacunar in a couple slides. And as you can see, we get different texture measurements depending on the scale that we're looking at with the plant. And so one thing that one of my students worked on for their thesis is we introduced this concept of something called lacunarity, which is a well-known texture feature. And so to make it a bit more concrete for you all, simply just measures the heterogeneity of your image. And so let's look at this top image here, okay? So we have like a regular, you know, checkerboard pattern here. So we have like a very low lacunarity value because it's pretty homogeneous, right? So it's not gonna have a very high value. But as you get very different patterns within your image, lacunarity can increase. And so this is a way that people capture this notion of like image texture or spatial arrangement in your image. And so from there, I mentioned that texture is very dependent on scale. And so one thing my student looked at was, could we incorporate some kind of multi-scale texture layer within a neural network? And so for those familiar with neural networks, very common pooling operations. So it's a way that you summarize or aggregate your information in your data, or average and max pooling. And so we did comparisons with those. And so this work was presented during the summer at CVPR, Computer Vision Power Recognition. So it's like a top vision conference in my area. And they had a workshop dedicated to agriculture. And so she was able to present her work there. And also our code is available for those who are interested also. Okay. And so also this image is the same here, just showing that our approach is able to capture a bit more information as opposed to the other pooling approaches that we compared with. So also along the image features, we can also look at different vegetation indices. So NDVI is a very common indice, so normalized difference vegetation indice. And so simply you're just looking at the near infrared and red information. And so we're able to get an NDVI measurement where it's bounded between 1 and -1, 1 being more healthy plant, -1 being like a dead or unhealthy plant. And so this example that we can also collect different vegetation indices in the facility. All right, so we talked about a lot, right? So we go from data collection, pre-processing to data analysis. And so how do you actually want someone to use this in practice? That's something that we're working on, right? And so I think everybody has a smartphone, right? So we have a bunch of apps on our phone. And so now we're working on developing like an app that people can use for our pipeline that we're developing. So very simple interface, wanted to make it very easy. And so someone just selects, oh, you just want to analyze plants. You say like where your files are located from the facility. And then we do everything that I mentioned before on the back end and then serve it to like an end user. And so as a part of this, someone can walk through and look at different visualizations. So in this example, we said, let's look at plant height. And one thing we want to make sure is that we're able to establish trust between an end user. And so we say we compute plant height. Someone can look at the max and minimum plant height for different plants just to make sure, okay, this software is actually capturing what I want to see. And so this is something that we added as a visualization aspect of this GUI. And also, you're very easily able to extract different plant statistics that we talked about, like height, width, and also like the number of leaves, different things like this that we discussed before. So also, I think I saw also on another slide, we also try and have good documentation too. So for those who are interested, you go to my lab website. We have this linked as well. And so it's a public repo. You all can look at the tool. It's available to use not only to people at A&M, but anybody who's able to access this tool can use it. And so, yes, we have the installation, just documentation we're working on. It's still an ongoing process. I think, like Noah mentioned before, it's really good to have good documentation. It's something that we're still working on with our group as well. So for the app, one tool that we use is Gradio. So this is a very popular tool, especially for developing apps for machine learning applications. So within Gradio, there wasn't support necessarily for multi-user, easy support for multi-user. And so one thing we looked at was using this ngrok, which allows people to use it like through a web interface and also, also locally as well. And so related to that, back in the spring, we had a chance to lead a workshop in the Ag and AI conference. And so I definitely encourage y'all to come next year. I think it's in Mississippi State next year. And so we had a chance to actually demo the workshop. And so for those who are interested, definitely be happy to talk offline. We have a notebook if you're interested to walk through the tool that we developed. We have a like Google Colab notebook that shows all the steps with this. And so part of this pipeline is not only developing research in a useful pipeline, but also graduate student development. And so really had big help from Yash and Uday, two former graduate students in my group that have graduated now. So another thing that we look at too is we want to make sure this tool works pretty fast, right? And so we look at the different sets that we talked about. Currently, the biggest bottleneck with our approach is really like the stitching and height estimation. The stitching is really dependent on like how many looks you have at the plant. So you can imagine as you take more images of the plant, you're going to have more constraints in terms of the image stitching that you're looking at. And so that's one thing we're trying to look at now is trying to speed that up a bit, as well as with the height estimation as well. But generally speaking, we have pretty good time frames. So starting off with like 7 plants, you can roughly estimate or analyze those in about 12 seconds. Okay. And so with that, along with the time analysis, one thing we're working on is that we're trying to develop this pipeline for real-time processing. So right now, a lot of it's offline processing. And so we're currently looking at something called Apache Airflow, which is supposed to help us like look at our different modules and aspects for executing our pipeline, right? And so if something fails in the pipeline, we want to say, oh, hey, flag us and let us know that we need to like work on and improve this like module that we're looking at. And so one of my students currently has been looking at this, and so we're hoping to make some more progress with this. All right. So I threw a lot of information at you all. So big takeaway is that we're developing a pipeline for the facility at A&M, and hopefully can be generalized as well. We're using tools such as PlantCV and existing approaches to help leverage this pipeline. Also, like again, I want to mention that a lot of this work is done by the students. So I want to thank Akshata and Yash in particular. Their work with their master's thesis really related to this work and really drove this approach. Also, this has been featured on some news outlets as well. So if you go to my lab website, it's been featured on PBS and Texas A&M Today. So you can also check out more about the facility as well. All right, and so with that, just want to thank Texas A&M AgriLife for providing support for this. Also want to thank the Plant Growth Phenotyping Committee. So I meet with them once a month, so they give me good feedback about how to improve the pipeline. Also want to thank my not only great collaborator, but also a great mentor, Dr. Seth Murray, for his help with this as well. And also just want to thank my students. And so for those who aren't familiar, at Texas A&M, we say gig them, so you got to give a thumbs up. So that's what we're doing here. But with that, if y'all have any questions, let me know. Thank you.